Hello, my name is Matej Martinc, and I will be presenting a study titled COVID-19 Therapy Target Discovery with Context-Aware Literature Mining. This is a study conducted together with my colleagues Blaž, Škrlj, Sergej, Pirkmajer, Lavrač, Bojan, Cestnik, Marzidovšek, and Senja, Pollak. So let's start with a quick introduction into the problem we're trying to solve. So there's this disease called COVID-19, which first appeared in late 2019 in China, and it's caused by SARS-CoV-2 virus. And a couple of months later, a pandemic was declared by the World Health Organization. And if you're at home, same as me, and not in Greece right now, you're probably aware that this disease is still wreaking havoc around the planet. So, of course, the scientific community reacted immediately, and COVID-19 instantly became the most researched topic in healthcare, and a lot of literature on the topic of COVID was produced. So I guess the best way to beat this virus is development of a vaccine, but as you know, this takes a lot of time, so sort of faster methods to deal with the virus are needed. And one way of this is to identify which antiviral drugs and targeted therapies developed to treat other viral diseases might be effective against SARS-CoV-2. And this is kind of the problem that we're trying to solve here. Basically, we're interested if we can discover some hidden latent scientific knowledge in the scientific literature about COVID, or to be more specific, we want to know if we can discover new COVID-19 therapy targets by using the targets discovered in the past research as seed concepts. These therapy targets I'm talking about here are proteins or genes to which SARS-CoV-2 virus binds to, and basically we want to know if we can discover some of these proteins that we can target with existing drugs in order to stop the replication of the virus in our cells. So first thing we did was look into the related work, and we found this study by Tian, where word-to-wake embeddings were employed on a large corpus of scientific papers to show that latent knowledge regarding future discoveries is to a large extent embedded in past publications. And we also then looked into the related work on text representation embeddings, as you're probably familiar with. First, word embeddings, like for example, word2vec produced one vector for each word, and they sort of ignored the fact that one word can have more than just one meaning. On the other hand, these recent developments like ILMO and very well-known BERT model produce different word vectors for different contexts of each word. So here we're talking about contextual embeddings. We also noticed that studies that would employ embeddings for discovery of new knowledge about COVID-19 are almost non-existent, or at least they were at the writing of this paper. And if they do exist, they're not peer-reviewed yet. So let's go to the methodology. So the data set that we're using in our study, and in which we try to find this latent knowledge, is called COVID-19 database, and it contains 135,000 articles about COVID-19, SARS-CoV-2, and related coronaviruses, and it's also freely available. And in this study, we also employ two types of embeddings. First are FastText embeddings, which we use as a baseline, and these are so-called static embedding, which produce just one vector for each word. And then we also generate text representation by employing SciBERT, which is a version of BERT pre-trained on a sample of more than 1 million scientific papers from Semantic Scholar, and is therefore more adapted to the task at hand than BERT, since it's already sort of adapted to scientific language. So the first thing we do is we fine-tune SciBERT as a mask language model on the COVID-19 dataset in order to adapt it to the COVID domain. And next, we extract contextual embeddings from this fine-tuned model as described in this image below. So basically here you can see BERT architecture, which contains 12 encoder layers. And basically what we do, we take last 4 out of 12 encoder output layers, and we sum them together in order to generate this so-called sequence embedding, and we cut this sequence into pieces in order to get embeddings for each word in this sequence. Okay, so at the end of this extraction process, we sort of have one contextual embedding for each word occurrence in the corpus, right? And then we aggregate these contextual embeddings on the token level. To put it differently, for every token in the corpus vocabulary, we create a list of all their contextual embeddings, and we average them. We average this list. So this way we get one representation for each token in the vocabulary. That is, we get one static embedding for one word in the vocabulary, and which means that we basically generate static BERT embeddings from contextual BERT embeddings. Next, we leverage semantic similarity to derive new scientific knowledge from the existing one. And in order to do that, we need some initial seed concepts, which are therapy targets, right? So for this, there are two possibilities. We can get seed concepts which are recommended by the expert. And this is what we did. We asked the medical expert to point out some seed concepts, and they recommended ACE2 and TMPRSS2. These are two proteins to which COVID-19 binds to and have a known confirmed link to the COVID disease in the literature. And another option to get seed concepts is to find them in the literature. And we looked at this study by Boykova et al., which identified a set of 2,802 COVID-19 therapy targets, which are also ranked according to the increase or decrease of production of a specific protein 24 hours after the infection. Out of this list, we use just the best ranked candidates, seed concepts. I will explain later how exactly we do that. Okay, so next we calculate embeddings for these seed concepts, and then we just look for semantically similar concepts in the COVID-19 dataset that are the closest to seed concepts according to the cosine distance between the embeddings. So this gives us a ranked list of new possible therapy target candidates, and we also filter this list according to the normalized Riemann distance to remove variations of the same base concept. Right, so this is basically our whole methodology, and now let's look at the results that we got. So the first way of evaluating our method is a so-called quantitative evaluation, where we use just two seed concepts. And here we evaluate if therapy target candidates acquired in the previous step have been confirmed as targets in the study by Boykova et al. To do that, we measure precision and recall at two different cases. One is 100 and one is 2,802, because this is the length of an entire list in the study by Boykova. And we also have two matching criteria. First is the exact match, and the other is a so-called fuzzy match, where basically we just, where two proteins match if they have the same beginning, and by this I mean if all the letters before the first number are the same, then there's a match. This method was sort of proposed to us by medical experts, and in this way we sort of get to match between genes that belong to the same family. Okay, so the results of the quantitative evaluation are presented in the table below. So on the left, you have results for SciBERT. On the right, we have results for FastText. And we also employ two different settings. In the first settings, we use two seed concepts proposed by experts, ACE2 and TMPRSS2. And in the second method, we use two seed concepts that were best ranked in the list by Boykova et al. at all. These were UBA2 and NCKP1 proteins. And as you can see from the table, SciBERT works better than FastText in all settings and according to all criteria. And out of these two settings, the better one is the one where we use UBA2 and NCKP1 as seed concepts. Here according to the fuzzy match, we get to precision at 10 of almost 50%. But the recall when at K is 100 is very bad, and we can drastically improve the recall up to 25% when we increase the K. In K is 2802, but this is at the expense of a worse precision. Okay, another experiment we conducted is we sort of try to find the optimal number of seed candidates. And this is exactly what this graph shows. So on the x-axis, you have a number of seed candidates, and on the y-axis, you have the recall. And as you can see in this graph, at least for SciBERT, we get the best recall when we have 32 seed candidates. These are 32 proteins that were ranked as the best in the list by Boykova. And interestingly, for FastText, you can see this distinct spike at the beginning when 4 seed candidates are used, and we're not exactly sure what's happening here, but we hypothesize that this is the result of FastText also employing morphological similarity besides semantic similarity, which is something that SciBERT does not do. Okay, then we also conducted qualitative evaluation, and here we generated 2 distinct therapy target candidate lists. First one containing 100 closest neighbors to the protein ACE2 according to the cosine distance, and the other one containing 100 closest neighbors to the protein TMPRSS2. And we gave both of these lists to a medical expert, and they basically what they found is that 9 genes, proteins, which were not linked to COVID-19 in previous studies, but appeared in both of these lists. And they researched them, and they came to this hypothesis, which is that indirect evidence supports a notion that phospholipases and prostaglandins, which are 2 families of proteins, might represent a target for treatment of COVID-19. You have a more detailed result and explanations in the table below. Okay, so let's go to the conclusion. So we can say that results of the manual qualitative evaluation by the expert indicate that at least 2 groups of novel therapy target candidates have been discovered. We have also showed that the proposed method outperforms the baseline FastText method by a large margin. On the other hand, the quantitative evaluation still indicates that precision and recall of the methods are relatively low in most cases. So this is one of the problems we will try to address in the future work, where we also address other issues that have arisen in the studies, such as gene synonym resolution and how to improve the pre-processing steps. And another thing we will do is we will try to find other possibilities of construction of meaningful semantic representations, because generating static embeddings from contextual one is not the only option. And we also plan to test other models, such as, for example, BioBERT. That's all from me. Thank you very much. And of course, I will be available for questions. Thank you.